Well, all right. Hey, Living Environment fans, we're back again on a Monday for another Bellringer. And this week, when it wraps up, we go on our winter break, which is pretty exciting. And don't forget, Thursday of this week, we have our next test, which will be on DNA replication, the cell cycle, and then what we're studying now, which is transcription and translation. So why don't we get to it now?
All right.
Hey now.
Mr. Kovacs class. It's Mr. Kovacs class. You might learn a ton. It's Mr. Kovacs class.
It's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class.
Remember, in DNA, A goes with T, G goes with C. T goes with A, G goes with C, C goes with G, right? Of course, it's different in transcription, right? Also, what else? When we do replication, we get 2 strands of DNA. When we do transcription, we get 1 strand of mRNA. Transcription, replication, 2 different things going on. Let's look at transcription and then its partner, translation. Transcription, right? You take a gene, which is a segment of DNA, you open it up and you copy it. The copy is called mRNA, messenger RNA, and this happens in the nucleus. Right? For sure, in the nucleus. That's transcription. Right? Transcription. Copying a gene is transcription. Then that piece of mRNA has to sneak out through a pore in the nuclear membrane to get into the cytoplasm before translation can happen. Translation needs a ribosome. Right? And a ribosome is kind of like, it attaches to the mRNA molecule and it keeps things organized. And then translation also needs tRNA molecules. tRNA molecules, which are floating around in the cytoplasm carrying amino acids. And what the ribosome is going to do is it's going to follow directions on the mRNA gene, or at least the mRNA template, and it's going to use those instructions to put amino acids together to make a protein. The building of a protein is called translation. It happens at the ribosome. You need a piece of mRNA, and you need tRNA as well in order to do translation. So translation makes proteins. Proteins, as you remember, are polymers. Their monomers are amino acids. There are 20 different amino acids that you can put together in any combination. It's like beads on a necklace, right? And you put them in combination and they make proteins. You put them together in a string, but because the amino acids are charged, they're polarized or hydrophilic or hydrophobic, the string bends on itself and folds and rebends and folds, so that when you're finished, your protein is actually this big 3-dimensional shape. It's kind of like one of those 3D printers, right? Like it prints just a line, but then you wind up with this total shape when you're done. Right? That's what happens during translation. It's the 3D printing of proteins by the ribosome. Today, what we need to learn is the code. Right? Learn the code. How does you know which amino acids put on next? And so we're going to talk about codons, anticodons, and how to read them. It's not really that complicated because, like most other things in life, if it was too complicated, it'd be too hard to continue. So, like life on Earth, ultimately, it's fairly simple. It's simple things that working all the time because they're easier to replicate. All right, so we'll do the Bellringer, do today's Ed puzzle, which is on translation, and then, we're going to learn the code.